Thank you for everyone joining today. For those of you who don't know me yet, I'm Alex from the Molecular and Genomics Informatics core facility here at Rutgers. We also have online Pat Soteropoulos from our Genomics Center, who handles all the single cell and the sequencing that we'll be talking about today, which is great that we have the Honeycomb team online who'll be talking about their specific platform for doing single cell sequencing. But before I turn it over to them, just the general housekeeping items. If you have any questions, please drop it in the chat or the Q&A, and we'll try to answer it live if possible. If not, at the end, we'll get to all the questions that we have. So with that, turn it over to Mike and Robert, the Honeycomb team, and let's hear about your awesome technology.
Great. Thank you very much, Alex, and thanks, Pat, for inviting us to speak here. This is going to be exciting, and I'm really excited about the Honeycomb technology. It's a very interesting way to do single cell RNA sequencing. So we'll get into that in a few slides. But just quickly, my name is Mike Bernan, and I'm the single cell product expert for PerkinElmer. So I'm global, so I cover, anyone that's interested in the HIVE. I'm happy to speak with them, and we'll see how the HIVE technology can support their projects. And I also have Robert Burden here as well. So Robert, you can introduce yourself.
Yeah, hi everyone. So I'm the local representative for the New Jersey, New York area. So I will help support all of you. If you're interested in trying the HIVE, I can help you with that, and kind of just help support Mike with all the fun logistics stuff.
Yeah. Well, thank you, Robert. So let me get into the slides here. I can kind of start outlining the technology, and feel free to drop questions in the chat, or we'll have some time at the end that we can kind of run through them. So just make sure I'm presenting. All right. So just to put together a little bit of an agenda for today. So I'll kind of go into like who is Honeycomb and what is the HIVE. So I'm sure you're wondering, these guys are from PerkinElmer. What are they doing talking about Honeycomb? But I'll explain the relationship. I'll go into just outline a little bit about what single cell RNA sequencing is for those who aren't as familiar with it, just high level. And if you are familiar with it, please feel free to tune out during those slides. And I'll go through some example data sets, showing a little bit more about the HIVE and its capabilities. And then finally, I'll go through the workflow, just so you can actually see, how easy it is and the different, the flexibility within the technology. So just a little bit about who exactly is Honeycomb. So Honeycomb Biotechnologies is a single cell genomics company that was started in the Boston area. So the relationship with PerkinElmer is that Honeycomb was actually incubated by PerkinElmer physically at the Waltham location, so PerkinElmer's headquarters. And so it really kind of built a nice foundation for the synergies between the two companies. And so originally Honeycomb was started as a spin out of MIT and the Broad to commercialize the seqWell technology. So that was started in Dr. Alex Shalek's and Chris Love's lab at the Broad and MIT. And so with this partnership with Honeycomb and PerkinElmer, really being able to expand the way we deliver this technology on a global scale. So we do have global customers now, not just within the US and Western Europe, but also expanding into Asia and Australia. So to get in a little bit about single cell RNA sequencing, I won't hammer this home too much, but to give folks a flavor of what's going on here, we like to think of typical standard RNA sequencing as kind of a smoothie. So you get this nice average of all the genes that are expressed in your sample. But what you lack in this case is the resolution down to the single cell level. So that's where single cell RNA sequencing really comes into play. So we think about that more as that kind of bowl of fruit analogies to see exactly what goes into that smoothie. So here on the single cell level, we can really pick apart and understand what are the specific gene expression profiles of an individual cell. So not only can we then ID the individual cell, but then we can look at the unique characteristics that make it differentiated from its neighbor next door. So that's really the power of the single cell method, and really to give that high degree of granularity that you do miss out with some of the other bulk technologies. And so what I've done here is just kind of outline some of the four main steps that go into the single cell process. So here I've started with your actual sample. So In this case, complex tissue. It could also be blood. It could be a number of different things actually going in to the single cell isolation method for sequencing. But this is really one of the critical aspects of the method is the dissociation part. So being able to get down to your single cell suspension without really disturbing the cells too much and trying to maintain that that biological complexity that you get, in its natural environment. And so working with fresh samples is kind of the gold standard. So any sort of fixation or cryopreservation, manipulation of the cells could potentially alter the transcriptional profile. And then, give you not as clean data, not as high quality data once you in your final sequencing readout. So that's why that step up front is critical. And then once you are able to get down to the single cell suspension, the next really important step is to enumerate and assess viability of the cells. So there's a number of different technologies out there for counting your cells. So Nexcelom, for example, being one of the gold standards for cell counting and viability assessment. But that really dictates how many cells to then input into your next step of what we think of as the high throughput single cell RNA sequencing method. So that could be, for example, a droplet-based approach, like this picture is of InDrop, for an example, a technology out of Harvard. It could be a pico approach like the HIVE, like we'll get into in a few slides, hydrostatic chambers from Fluidigm. So there's a number of different ways that the cell gets isolated and then uniquely barcoded. And that seems to be, pretty standard across these different technologies, regardless of how you're isolating the cell. And then once you've built your library and sequenced, the last step of the process is data analysis. So if you're not familiar with these type of plots, in this case, each individual dot represents a single cell, and then you can cluster based on a number of different factors, such as cell type, for example, to understand what are the different cells in your population. And then kind of dive into the complexity of your data from there. So some of the major limitations of the current single cell sequencing technologies, it really comes down to, access to your fresh samples is kind of the crux of a lot of these technologies. And some part of that being the actual complexity around the collection of these different samples. So typically you need specialized equipment. So, you'll need an actual instrument to do your single cell isolation and barcoding. You'll need, cell counting. You'll need all these different pieces of the puzzle to complete the process. And that's just not feasible in some of these different, either it's a remote collection site or if it's a lab that's not as well funded, that doesn't have capital equipment budget. So that really limits who can actually do the single cell sequencing. Another method or another limitation, is these different storage aspects of how to preserve your sample. So like I mentioned before, using fresh sample is kind of the gold standard, but there's a lot of these other methods out there that folks like just kind of fall back to because they aren't able to use their fresh sample for their single cell sequencing projects. And then another one being some of the other approaches, some of these droplet approaches, they are quite harsh on the cells going through. So, shoving a cell through a small microfluidic chamber really exerts a lot of shear stress on the cells. And these shear forces can potentially lyse the cells, make them unhappy, increasing mitochondrial reads. So there's a lot of limitations there that come with droplet-based approaches. And one of these, the key ones being the actual instrument itself. So if you are working in, for example, a BSL-3, BSL-4 facility, then you'll have to have an instrument dedicated specifically for that facility, and you don't have the flexibility of taking it in and out. And along the lines of the harshness of some of these approaches, being able to recover certain fragile cell types like granulocytes, for example, and I'll run through some data around those tend to get lost in whether it's cryopreservation or some of these droplet-based approaches. Sometimes you're not able to recover as much diversity in your population as you would expect. And so to get in a little bit about the HIVE solution and how it works. So I've kind of laid out the general steps here for the method. So you can see in this picture right here, this is the hive. So it's I wish I had a scale, but it's pretty handheld, small device, about 1.5 inch by 1.5 inch. So it's very portable. And really it was specifically developed for distributed studies. So being able to take this collection device to the site where the sample is being collected, and loading the sample and preserving the sample in the device, and then sending it back to another facility for processing and sequencing. So because we want to really get into these distributed studies, we had to make it instrument free. So it really all you need is the device, and then some reagents and standard lab equipment to process the libraries, and that's all you need for building a sequencing ready library. And like I mentioned before, with the droplet-based approaches, because we are using a picowell system, and the cells are loaded either by gravity or by light centrifugation, we actually maintain and conserve these fragile cells quite well. So we're not exerting them any shear stress on these cells. So we're able to get a really nice picture of all the different cell types in your sample without potentially biasing and overrepresenting certain cell types. And then I mentioned before the actual storage in the device. So we are able to add a stabilization solution. So this is essentially a high salt buffer that precipitates genomic material and allows you to store the loaded cells in the device here for up to 9 months. And so this isn't a fixation method. It's more of a preservative. So we're not cross-linking RNA or anything like that. And so we're still able to do a whole transcriptome capture at the end of this. So this is kind of the general process at a high level of the hive. So we start with our single cell suspension. We load the cells directly into the device. We add our preservation solution. And then at this point, you can store in the device frozen for up to 9 months. So you could ship the samples, anywhere you'd like, or you could bank them in your own freezer and then pull them out when you're ready to process. And when you are ready to process, you go through, you'll lyse the cells, you'll capture the mRNA. I'll get into that in a few slides. And then you build your Illumina-ready libraries, sequence, and then do your data analysis. So to show a little bit of data around, specifically around the storage component. So that's really one of the unique features of the Hive that no other technology is able to do, is to the ability to store samples and then either ship them or bank them and process them at a later time. So it really gives you a lot of flexibility, especially as a core facility. Not having to worry about the logistics of when you'll be able to process these samples. Will you have to stay up till 10 p.m. to build these libraries, or at least get to the cDNA synthesis step. So it really gives a lot of flexibility to how you design your experiments. So in this specific study, we looked at the Hive versus cryopreservation. So we wanted to see, with our storage method, are we potentially losing things, or how do we stack up against kind of the current standard of cryopreserving samples prior to running single cell sequencing? And so in this study, we looked at healthy human bone marrow. We did a red blood cell depletion. So the reason we did a red blood cell depletion using the stem cell tech RBC kits was really we want to preserve granulocytes. So we know that they're fragile. We know the Hive can be great with fragile cell types. So we wanted to see how those stacked up in the Hive versus cryopreserved samples. And then we looked at the Hive stored in the Hive for 1 week versus cryopreserved at 1 week. And then also we extended that out to 4.5 months and then 9 months. And so this is compiled data from all the different conditions. So both fresh, stored in the Hive for 1 week, and then cryopreserved. And what we really wanted to hone in here on is the neutrophil population. So this pink, purple, blue kind of cells. And so, we know that they are very fragile. So we want to, hone in on them for each of these different conditions to make sure that we're still maintaining these fragile cells, across the storage, and then how that compares to cryopreservation. So in the fresh, kind of as we expected, we saw the large neutrophil population, this kind of kidney bean shaped cluster, and then stored in the Hive. for 1 week, we still were able to maintain the diversity of that neutrophil cluster. And then when we compared that to cryopreservation, we essentially lose that entire cluster, and we see the emergence of this smaller cluster down here. And we kind of dove into this cluster and realized it was the neutrophils that were able to be recovered that didn't get too beat up with cryopreservation, but their transcriptional profile is changed in such a way that they don't even cluster like their like the other conditions. So we asked the question, how does that are we really mimicking the biological relevance of the sample using cryopreservation at the end of the day? And then we dove into different percentage of the populations these different cell types make up. And so looking at the fresh versus stored in the hive after 1 week, we saw, as we expected, about half of the sample being neutrophils for both the fresh and stored in the hive for 1 week, versus in the cryopreserved samples, you see the neutrophils here is less than 10%. So we lost the majority of these neutrophils, and then we see an overrepresentation of more robust cell types in this sample, such as T cells, sitting on making up more of the population than they did in the hive fresh versus stored in the hive for 1 week. And so we dove into the neutrophil population a little bit further, and we saw really nice, robust expression markers, and we're able to cluster based or able to find subclusters of these neutrophils. So we were able to find 11 different subclusters, which you're really only able to obtain if you are able to capture a large number of these neutrophils. So the fact that we were able to conserve these during the fresh and the storage process really gave us this granularity to look at this unique cell type. And then we want to make sure that we weren't potentially changing the diversity of these samples at all. And so we looked at the percentage at which these cell types arise in the versus the hive fresh versus the stored in the hive. And so this plot down here is just showing that when we look at these two different conditions, stored in the hive after 2 weeks versus fresh, we see pretty much the same fraction of cell types for the two conditions across these different clusters. So it gave us confidence going in to further studies that we weren't compromising data quality by storing these cells in the hive. And so we ended up extending the time points, because typically a week or 2 weeks is not enough for storage. So we ended up extending this out to 9 months. And so we did the similar type of analysis where we looked at the neutrophils and eosinophils and basophils. So this cluster right here, we looked at that in the fresh, then stored in the hive for 4.5 months, and then stored in the hive for 9 months. And we see, good conservation of this cluster across these different time points. So really giving us confidence that we're not missing any of the granulocytes by storing for this long period of time. And so finally, we looked at the gene expression profile of stored in the hive for 9 months. We really want to make sure that we weren't, not only were conserving cell types, we weren't changing the expression profiles. And you can see here in the orange is the fresh versus the black is the 9 months. So we really didn't see too much of a difference in the actual expression profiles from these different storage conditions. So it's really nice to see that, gives us confidence moving forward that we might actually be able to extend the 9 months. We are working on 12-month storage, but also we're not compromising data quality, because that's, at the end of the day, we want to make sure that you're able to confidently use this method and not have to compromise any of your experimental design. So in this next study that I want to share, this was along the same lines of that preservation study I just shared, but it was more of a real world example. So this is a collaboration with one of the top 10 pharma companies over in Europe, and they really liked the hive because of the multi-site collection aspect, as well as the fact we're able to pick up granulocytes. And so in this pilot study, we used whole blood and liver fine needle aspirates from 3 healthy donors. And these samples were collected and loaded in the Hive at different clinical trial centers across Europe. And they were all shipped back to Honeycomb in Waltham, Massachusetts for processing and sequencing. So the entire logistical time at which these samples were stored was about 2 or 3 weeks. And so this is the data that we saw from the blood and the fine needle aspirates. So we're, essentially the company was very happy to see we recovered pretty much all the expected cell types that they're after. And they really were excited about the fact we saw this large granulocyte population. So in the bottom here, in the orange, yellow, green kind of cluster here. So typically with their standard method of cryopreserving these samples at these different sites and then shipping them back to their headquarters and processing and sequencing, they lose the granulocytes. So they're very excited to see that, especially because they were working on specific autoimmune and inflammatory diseases where granulocytes play a big role in. So it was very important to their work to really have a nice understanding of the granulocytes at the single cell level. And so this is the gene expression profile from the data. So we saw really nice transcriptional heterogeneity between the neutrophil subclusters. So these 4 different subclusters of neutrophils, as well as we were able to see some hepatocytes as well from the fine needle aspirates. And this is tricky because hepatocytes typically aren't able to be recovered from fine needle aspirates, just the way based on the way the biopsy happens. And what I thought was the most interesting part of this study was when we compared again that percent of the population breakdown based on cell type. When we look at whole blood, we see basically half the population is neutrophils, as we would expect, and then a smaller percentage being CD8 cells and a smaller percentage being cytotoxic cells. But then when we look at the fine needle aspirate data here, we see 30% being neutrophils, which is actually really nice to see that we were able to detect tissue resident neutrophils. So that was exciting. And a larger percentage of CD8 cells and cytotoxic T cells, but it was this emergence of a tissue resident NK cell population that was quite interesting as well. So doing this kind of pairwise comparison between these two sample types, really kind of gives you a lot of unique information if you are looking at from a potential biomarker perspective, or if you are looking at immune cell infiltration based on a given therapeutic modality. There's some really interesting type of information you can gather from these two kind of pairwise sample comparisons. So it really bode well the fact that we were able to get this type of resolution and granularity with the flexibility of collecting these sites, across Europe and sending them to a single processing center, processing and then sequencing. So up until this point, we talked a lot about kind of blood and more fluid based single cell sequencing, but I did want to share this study that we did with a core facility in Australia using resected glioblastoma samples. And so the glioblastomas were resected and then dissociated with Miltenyi dissociation kit, and then the samples were stored and then processed on the Hive. So this is just kind of the general workflow of what happened. So after resection, so it was specifically the GenoMax dissociator that was used, and then the cells were loaded on the Hive after right after processing, stored for about a week. And then once that they were ready to process, and it was more for convenience sake, it wasn't really for any biological purpose. It was the core facility was busy, and so they decided after a week they would take it out of the freezer and process it. They built the libraries and then they sequenced. So about 20,000 cells were loaded per device, which was really nice to see. So not out of a huge amount of cells. We were able to see up to 16 different cell types. So, and almost half of them being malignant cell types. So it was really interesting to see, some of these immune cell populations within the glioblastoma sample. So that are quite challenging to see with other methods. So, just in general, really robust expression markers across a lot of these different cell types. Was really, really interesting to see. And if you do want a little bit more information on this, we do have an application note as well. So it's on the Honeycomb and PerkinElmer websites. So again, we did this kind of breakdown of different percentage of the population that are of various cell types. And, we were over 50%, so 68.5% were malignant cells. So it really gives you an idea of the frequency at which these malignant cells arise, and then how does how does that affect the rest of the biological diversity of the sample? So the fact that we're able to capture this wide swath of diversity, anything from fragile cells to these more robust malignant cells, really gives you a nice picture of biologically what exactly is going on in your sample. And so this data set that I want to share was, looking at the HIVE versus a droplet microfluidic method. So I can't say exactly which droplet microfluidic method, but you might be able to infer which one it is. So this was done on healthy human blood. We did a red blood cell depletion, and both the HIVE and the droplet method were loaded fresh. And so the kind of the point of this study was to not really say, which technology is better, but it's more along the lines of, what technology fits your biological question better. So in this specific study, we were really focused on, again, the fragile cells, so specifically granulocytes, to really understand, we understand the HIVE can see them, but do they get missed with some other methods? And so, to help guide folks along the single cell sequencing journey, as to advise them, if you do have a project that you are really interested in cell diversity, or you're really interested in some of these more fragile cell types, or your samples do have fragile cells because you have done another method and you don't see them, that's where the HIVE really can come in and kind of fill that hole. So in this specific study, this is the UMAP plot from the whole blood data. So we again honed in on the granulocyte data here, so this kind of bird head shaped cluster. So we were able to see a good chunk of them in the HIVE data, which is really nice to see. And then when we look at the droplet data, we really don't see any of the neutrophils or granulocytes in general. And so we thought, we'd highlight based on the specific features of the neutrophils and granulocytes. And again, we still didn't really see too many in the droplet method data. And so when we kind of broke down based on percent of the population that these granulocytes are, again, in the HIVE, about almost half were granulocytes from whole blood, which is, again, what we'd expect, versus in the droplet method, less than 1%. So it's, clear that, the droplet method, it might be great for some things, but, if you are interested in these more fragile cell types, then you might be missing some of the some diversity in your sample. And so just for reference, we thresholded our data based on about 400 genes and 800 transcripts. And we kind of took this, we decided to see if maybe we're filtering out some of the granulocytes in the data. And so what we did was we actually lowered the threshold to 50 genes and 100 transcripts in the droplet method. And still, we don't see great, robust expression profiles from the granulocyte populations. So we came to the conclusion that, they just weren't there. It's not that we were filtering them out or anything. There was something with whether it could be the fluidics involved, or something they were potentially pre-lysing before they even got into the droplet and were barcoded. So now to go into a little bit more about kind of the HIVE, and I know I have about 15 minutes left, so we'll kind of run through a little bit about the workflow, and then we'll save some questions at the end for if there's any Q&A. And so kind of now that you've seen some of the data around the HIVE and some of the benefits of, for example, the storage component, or capturing really good cell diversity, you can see how this could be potentially deployed in a multi-site study, for example. So we do have some folks, for example, in South Africa, collaborating with folks in Boston, where they're capturing samples in more rural villages in South Africa and sending them back to Boston or sending them to their local core facility for processing and sequencing. So it kind of really opens up how you want to do your single cell experiment. You're not really limited to either cryopreserving or having some unique instrumentation on the site where you want to do these experiments. And so, unlimited collection sites is definitely one area that a lot of users value. Being able to collaborate easier with whether it's across the pond or across the US. These different time course studies. So being able to bank these samples for right now 9 months. We are potentially extending that soon to 12. But just giving you that freedom to kind of pull samples out when you are ready to process and sequence them, and not have to, worry about the challenges that come with getting a sample in at a certain time, processing it up to a certain point, and then stopping. It's, sample comes in, dissociate, load Hive, preserve, and then store. So this is just some of the different sample types that have been done in the Hive successfully. And the Hive was just launched about a year ago. So we are still pretty new to the market. And so this list is growing on a weekly basis. As new folks get the Hive, as new folks try out these different sample types, this list is growing. So if there's a specific sample type that you're interested in, please let myself and Robert know, and we're happy to try and connect you with someone that's done something similar or share our experiences with that specific sample. So once you've kind of have your sample, and you go through that tissue dissociation, or getting down to your single cell suspension, and you've counted the cells and assessed viability, the process is loading into your Hive device. There's a quick wash step, and then preserving your samples for processing, at a later date. And so the reason I mentioned the Nexcelom Ceilometer here is we do actually have an app note with Nexcelom that we recently launched. So if you're interested to learn a little bit more about their cell counting capabilities, you can check out the website and see the app note there. And it really kind of shows nicely how trypan balloon might not be the best way to count your cells for single cell sequencing. So once you've now preserved your samples, so you have 9 months before you are ready to process them, or before you have to process them. But that being said, once you've loaded the Hive, you don't have to store them before you go into the processing. If you want to, you can go straight into the processing. And so the next step here is you'll seal the Hive itself, so the array. So there's a little membrane that you seal the array. So this seals the wells off from one another. You add your lysing solution. The cells get lysed. The mRNA gets captured, and then you recover the beads. And it's a fairly standard library preparation. I added this filter plate here because in some of the steps, your mRNA is still attached to the bead. So, instead of just pelleting and taking the supernatant or doing a mag bead cleanup, we wash the beads over a filter plate to clean them up for the next reaction. And then finally, that'll create a Illumina ready library that you can then sequence. So to give a little bit more visualization of what exactly happens in the capture devices, so this is kind of the general steps for loading the device itself. So you'll thaw the Hive. So you load your cells. So it's one sample per Hive. We can load anywhere between 500 and 30,000 cells in 1 ml, or between 1 and 4 mLs. So the Hive itself can hold up to 4 mLs. So you can either load if you have 4 mLs worth of cells, you can load all 4 mL, or you can load them in 1 mL and just top the rest of this Hive up with media. So once you've loaded your sample, you'll go through and you'll either spin the cells down in a centrifuge, or you can let them sit for 30 minutes at 4 degrees, and the cells will settle into the picowells. So on the bottom of this hive, there's 65,000 of these picowells. And so this is just a cross section of the picowells. So in each picowell, there's this barcoded bead. And on the barcoded bead is decorated with these oligonucleotide barcodes. So it has, your cell barcode, your transcript identifier, PCR handles, and DT capture sequence. So the cell will settles into the well here. And then when we add in the preservation solution, so this locks in that transcriptional signature of the freshly loaded cell. So that's what gives us the ability to then put it in the freezer and freeze it, or ship it to another location. So when you are ready to process, this is just, I thought I'd go through one of the first steps here, because the rest of the downstream is fairly standard library preparation. But what you'll do here is you'll pop the top off here. You'll add on the membrane. So this is, as you can see in this picture here, it's a semi-permeable membrane. So it allows small molecules like what's in our lysing solution to flow through, but it won't let any large molecules like nucleic acid diffuse out. So this will specifically heat seal on, and it'll trap the cell in the well with the bead. So once the cell gets lysed, we don't have to worry about any potential cross-contamination between the wells, because everything's sealed in its own individual reaction chamber. So the nice thing about a picowell technology versus a droplet approach is we do have more flexibility with our lysing conditions. So we don't have to worry about putting another surfactant into a droplet and having that behave with enzyme XYZ. So we can do buffer exchanges with these picowells. So our lysing conditions are a little bit more harsh than other methods, and we've shown that it deactivates viruses, for example. So once you've gone through the sealing, the lysing, the capturing of the mRNA, and that's fairly standard library preparation after that. After the sequencing, we have our BNet software. So this is the back-end pipeline that'll take your FASTQ files, and it'll do all the QC and alignment, and it'll spit out a gene counts matrix. So it's a standard CSV file, then you can then upload into whatever analysis package you prefer. So it's compatible with R and Syrah and ScanPy. So we don't kind of limit you to using one specific pipeline, because I know everyone seems to have their own kind of favorite pipeline depending on what the experiment calls for. And this can be run locally. It can be run through Terra, hosted by the Broad. So there's a lot of flexibility in how you want to run the pipeline as well. So I thought before we start opening it up to kind of Q&A, I thought I would share a quick video of how the cells get loaded. So this is short video, but you'll see kind of how easy it is to run the method. So this is your hives. This is you'll get them frozen. So you'll let them thaw. You'll remove that little stopper, and then you'll remove the fluid that's in there. So that's just to keep the membrane moist during shipping. So you add your single cell suspension. So in this case, these cells are settled by gravity, but you can also put them in a centrifuge. So you'll remove that media. You'll wash the sample, and then you'll add in your preservation solution. So you put the stopper back in, and you can put it into that little box, and you stick it at -20. It can be stored at -80, and it can be shipped on dry ice. So that's the capture portion. As you saw, it's 6, 7 pipetting steps. So pretty straightforward, pretty easy to do. All right. So, in summary, just, kind of highlighting some of the points of the Hive and, why it's a pretty unique solution out there for single cell sequencing. Our ability to capture fragile cells to really preserve that diversity of cell types in your sample. To be able to store, these cells long term without compromising data quality or degradation of your sample. And then this enables a lot of these different stopping points along the way. So you really have more flexibility in how you want to design your experiment. And you don't really have to design your single cell experiment around, a specific piece of equipment. So if you'd like to reach out, feel free to reach out to myself and Robert. So these are our emails, and I will pass around these slides afterwards. And so if you do have any questions, you can reach out to us. All right. Thank you very much.
Awesome presentation. If anybody has questions, put them in the chat and we'll ask. But I have a couple while we wait for everybody to kind of formulate their questions. So just out of curiosity, I know you guys said that you have 9 months validated, you're working on a 12-month storage validation. Do you have any anecdotal people who've stored it for 2 years or 3 years or anything like that?
So since the system's only been out for a year, so okay. we have some our own lab here have stored it for at least 18 months, and it's been fine. But we need to further validate before we can officially say that. Yeah, but hopefully in about another 6 months we'll have some more users saying, "Oh yeah, I kept this for 2 years, and there's no problem."
Cool. Okay. Yeah, I always like to hear the anecdotal shenanigans that are going on. But then another question too. So you mentioned, or I saw on the one side talking about using BSL 3 and 4 settings, and that's something we do have a BSL 3 setting here at Rutgers. We have a lot of people that do a lot of TB work and various other viral works as well. Has the buffer has been validated for inactivating the samples?
Yep.
Oh, yep. Perfect. Okay.
Yes. So actually, I'll send you a link. Maybe you can pass it around as well. But we recently had a Genome Web webinar with one of our customers, Dylan Sheeran, out of WEHI, so Walter and Eliza Hall Institute in Australia, and he's working with TB samples as well as COVID-19 samples. So he's collecting them in in South Africa, and shipping them to Australia, and he's working in the BSL-3 facility, specifically looking at neutrophils. So it's actually a very interesting webinar. So I'll share that link with you if you want to. You can pass that around.
Yeah, please do. But then with that too, so if it's working with these viral samples, I know it's a limitation with some of the other technologies where they've had difficulty capturing lowly expressed genes. In particular, I know we've had a couple people try to capture it, whether it be flu, COVID-19, various other polyadenylated viruses. With that one you just mentioned, where they're doing COVID-19 in the hives, are they detecting the viral transcripts as well?
So I can't recall if in that one he did, but there are some folks that are able to detect they are able to detect the viral transcripts. It's obviously, quite hard, but with enough sequencing, it's possible.
Cool. Yep. Oh, that's awesome. Okay. Does anybody have any questions besides me? Oh, we got one coming in. So it says, so you said 30,000 can be loaded. How many are captured from that?
Yeah, so between 500 and 30,000 can be loaded. So the typical capture rate, it definitely varies sample to sample, sample quality, et cetera. But in general, our range is between 20 and 30%. So that's, again, could vary a little bit from that based on the quality of your sample, but that's kind of the general ballpark for recovery.
So I have a question. What types of different cell types have you tested? So, because people have so many different cell types that they bring to us, and every cell type needs, some different, QC, some different optimization.
Sure. So in general, as long as the cells can fit in the wells and can be lysed and have polyadenylated mRNA, they can be captured in the hive. So, I guess if there's any specific cell types or sample types that you are thinking of, I can send over in my slides. I had a bit of a list. I can actually let me just pull that up so everyone can take a quick look. Because even off the top of my head, I can't remember all the different cell types.
Yeah, I think you said it was changing weekly.
Yes, changing weekly. Yep. So this is just a small list. I think this is a couple weeks old, so I think there's a couple new ones in there. But anywhere from obviously blood is a definitely a popular one with the Hive, but also moving into more tissues and various tumors, and also maybe not, I don't want to say non-traditional, but non-mammalian organisms, I guess is the best way to put it. So we have another number of folks working with plants, as well as some funky marine invertebrates, echinoderms, looking at different stages of the life cycle. And that was interesting because we had to load them in a high salt solution, which the Hive fared well in. So it's got some nice flexibility as well in terms of what buffers the samples can be loaded in.
We have another question coming in. So what's the difference between Parse Biosciences, where they had the free sample prep, and yours is I guess because they have the different fixation buffer based on the barcodes. Kind of what's the big difference is what they're asking between this and Parse?
Yeah. So I'm not an expert in parses. I believe it's some sort of fixation. But so with theirs, as far as I understand, you're fixing up. Well, you're going through a fixation up front, and then the storage is potentially limited between when you fix and then when you actually have to process. I think it's only a week or maybe a couple of weeks. They might claim longer, but from what I've heard, there's some limitations there. But it's mainly that because we're not fixing the cell, so the cell is actually already isolated, and then we're preserving the cell. We're not worried about how the cell is going to react to that different fixation. So I know if you're working with a really heterogeneous sample type, you might have varying degrees of how the cells actually respond to that fixation, how well they're fixed, etc. So the fact that we're isolating them already, so if the cell kind of pre-lyses or anything like that, it's still all contained in the well. So we're still able to capture the transcripts from those cells. So I hope, does that kind of answer the question?
So hopefully, if not, they can follow up with more questions.
Sure.
But another question I have as well. So some of these other technologies are coming out with these different multimodal assays where you can capture cell surface protein markers and things like that. Is that compatible with the Hive as well?
Yes, that's a great question, and I should have mentioned that actually. So we are working with BioLegend. So now that BioLegend's part of PerkinElmer, it's a really nice collaborative effort between the groups. So we're working to get the TotalSeq A kits up and running on the Hive. And TotalSeq A, it's meant to be kind of platform agnostic. So it's more about formalizing an actual protocol around that. So we have verified it works, but now in order to support it as an application, we just got to put a more official stamp of approval on it.
Cool.
What about nuclei? Did you mention that and I missed it? But because I'm sorry, you might have mentioned it and it went past me.
Yeah. So nuclei can be done as well. It's the only changes to the protocol really is we recommend spin loading instead of gravity loading, just because nuclei are a little less dense than whole cells. But besides that, that's really the kind of the only difference between how you process them. And nuclei, I will, just to be honest, we don't see too much of a difference between nuclei on the Hive versus nuclei on 10x. It doesn't seem to be really much advantage from a sensitivity standpoint, or being able to capture fragile things or store the nuclei. So that tends not to be a problem anyways. But they can be run.
Okay. Cool. Does anybody else have any additional questions? Okay. Well, I guess then thank you for a great seminar, and thank you everyone for joining today. We'll send the recording out in the next couple of days, and Mike said he was going to send over the slides, so we'll send that out with it as well as their contact information. But if anybody else has any questions or any follow-ups, you can reach out to myself, obviously Mike or Robert, Pat in the Genomics Center, and we'll get you, hooked up with the appropriate parties so you can talk through your project. But thank you everyone for joining today, and look forward to seeing everyone next week for the next seminar.